Okay. Good morning, guys.
Morning, sir.
Good morning,
Morning,
sir.
sir.
Good morning, sir. Morning, all of you. Okay, guys, I hope you're able to see the screen.
Yes, sir.
Yes, sir.
Okay, let's go on. So we'll do measles virus. I'm changing the order of the lectures a little bit. It doesn't matter. So today what we're going to do is we're going to try and do measles virus, and there's a group of viruses called measles, mumps, and rubella virus that are always discussed together because they have similar pathogenesis or they cause similar disease. So are you vaccinated against measles virus? Yes, no? What do you think? Okay, most of you are, I think. If you've taken all the regular vaccines, you have been vaccinated against the measles virus. Now, a little bit about the measles virus. It's a negative sense RNA virus. Size is slightly bigger than the viruses that we've looked at so far. We've looked at the polio virus or the enteroviruses, which are picorna viruses, really small viruses, maybe about less than 15 nanometers in size. Then we looked at influenza viruses, which are slightly bigger, 80 to 100 nanometers. These are even slightly bigger. They're 150 to 250 nanometers in size. Now, measles virus has been one of the most contagious viruses that has been known to mankind. Of course, now we know SARS-CoV-2, and before that we knew influenza. But this is in that league of highly contagious viruses that actually threaten mankind. But we came up with a solution a long time ago, so that's why we don't talk about measles virus that much, but it is still a problem, and you'll understand by the end of this lecture why we are talking about measles virus. Now, what I have here is it also causes a rash that is called a maculopapular rash. I'm going to qualify what that is and tell you a little bit more. So these are some 4 terms. Don't get worried. They are very simple terms, but they're important in terms of understanding what virus infections mean. Now, a macule is just the discoloration of a skin. Okay, that's called a macule. It may be caused by a virus, it may not be caused by a virus, but it's called a macule if you have discoloration of a skin that has no elevation. But if you have elevated, like it's kind of, there is an elevation that you can see on the skin, but there is no fluid inside. That's called a papule. And then there is the next one is a fluid-filled elevation. It's called a vesicle. As you can understand, vesicles are filled with fluids. So if you have a fluid-filled rash, then that's called vesicular rash. Then the other last one is very easy to understand. It's called a pustule, and you get a pus-filled elevation of your skin. Now, maybe before we'll go on to measles, which causes maculopapular rash, perhaps if anybody can give me an example of when you see a pustule. Guys?
When you have pimples, probably.
Yeah, but what is inside the pustules? You think viruses or something else?
So I guess bacteria.
Good guess. Okay, pus is usually associated with bacteria. Viruses usually do not cause anything to do with pus. It's usually bacteria that cause pus-filled lesions. So when you have a pustule, it's usually a bacterial infection. Now, vesicular rash, fluid-filled elevation of the skin. Do you think viruses can cause it? Yes, no, guys, make it a little bit more interactive. No? If I remember right, one of you said, yeah, go ahead.
So I'm not sure, but maybe not the virus.
It can be caused by viruses. One of you, I don't know who, because I go in full screen mode, I can't see who's exactly speaking, but I remember one of you explicitly said that you had chicken pox after vaccination. Is that right?
Yes, sir.
Okay, if somebody got chicken pox after vaccination, the chicken pox rash should look like this. It should be fluid filled elevation. Okay, so the difference is here between measles and that. Chicken pox rash will have fluid, measles rash will not have fluid. That is the main difference. But in any case, this terminology is important because I remember I told whoever said that they had chicken pox, I told that one of the 2 times that they had chicken pox or they had it after vaccine, it could be a misdiagnosis because it's only a clinical diagnosis. That comes from the fact that there are other viruses that can cause similar rashes. So anyway, I just remember that while talking about macular-papular rashes. Okay, so measles causes only these 2 types of rashes, macules and papules, so it's called macular-papular rash. Humans are the only hosts, and like you can imagine that when I said this is one of the most contagious viruses known to mankind, there is no need to second guess. It is, of course, being transmitted by respiratory route and aerosol. All the highly contagious viruses that infect humans are transmitted only through this route. It replicates a little bit in the regional lymphatic tissue, especially the tonsils, and then it enters the bloodstream, and it can infect all the epithelial cells in the body. So where are these epithelial cells? Epithelial cells are in a lot of places. Particularly what you want to remember is that all the membranes, like on the inside of your nose, inside of your mouth, inside of your food pipe, windpipe, digestive tract, urinary tract, wherever eyes, all these, wherever you have moisture, it's all lined by epithelial cells in the body, and measles can infect pretty much all the epithelial cells in the body. So that's what it infects. So something different about measles in terms of pathogenesis, it has 2 distinct receptors. So this is one receptor, this is the other receptor. I'll tell you what it is very quickly. So when the measles virus, like I told you, it enters the body through aerosol or the respiratory route, it is able to replicate in the lymphatic tissue, particularly tonsils or other lymph nodes in the respiratory tract or the back of the mouth. And there it uses a receptor called SLAM, which is an important, it is a cell surface molecule that is present on T cells and B cells and is necessary for activation of T and B cells by signaling. So once it replicates here and spills over into the blood, it can seed all the epithelial linings, because then it uses another receptor, because they don't have SLAM. It uses another epithelial cell surface receptor to affect pretty much all the epithelial cells in the body, like any part of your body that is moist, including the airways, the lungs, the oral cavity, the intestine, and the urinary tract. They're all moist and they all have epithelial cells, and measles can infect that. Because it infects the lung and this part, the infected person, when they cough or when they speak, they bring out aerosol that contains millions of measles virus particles, and that's how it's transmitted to the next person. So what is different so far about measles virus? So far the main difference is it is a papular rash. The second thing that we looked at so far is that it can use 2 receptors. Unlike most viruses that we know of, it uses 2 receptors. Some are distinct in immune cells, T and B cells, called the SLAM, and then all the epithelial cells have another receptor. It can use both the receptors to enter human cells, and the transmission is aerosol. Okay, so you see this whitish spots on the inside of the mouth. So these spots are called Koplik spots, and this is the way clinicians usually diagnose measles. So this happens a few days after infections, but you can see that this child is also having a rash, but If you have a rash, that is not just enough. Even if it's maculopapular, it's not enough for the diagnosis of measles. But you see this characteristic white rashes inside the buccal mucosa of your mouth. That's called complex spots, and that's how most often it's diagnosed. Now, just the same cartoon zoomed in. So this is the maculopapular rash. You can see there is no elevation. There is discoloration of, sorry, there is no fluid. There is discoloration and elevation of skin. So that's just some minor information about clinical presentation. Now, if you get infected through aerosol somewhere here, let's call that day 0. It grows in your tonsils within about 2 to 3 days, spills over into the blood around 5th day. That's viremia here, 5th or 6th day. It can see the epithelial cells of the body within 1 week of infection. It replicates in the epithelial cells, and it can cause a respiratory illness because it replicates in the lung. That's about little over a week. When do these complex spots, which I showed you, when do they appear? They appear around 10 days of illness. You can see that. That's immediately after you have the respiratory illness. It's more or less matching with that. Before that, you only have rashes. No, sorry. Before that, the virus only grows. You don't know anything. Respiratory illness is the first presentation. Complex spots occur almost along with it. The rashes occur a little bit later. So what is the blue line here? The blue line here is the amount of virus in your blood. It keeps on increasing. It plateaus a little bit and it decreases. And the rash appears somewhere around this point when the virus load is decreasing and when the antibodies or the immune response is increasing. That's when the rashes appear. It kind of coincides with that. Now, anybody wants to think what could explain the rash in measles virus infection based on this cartoon? At least make an informed guess.
So our own immune response.
That's correct. It's mostly antigen antibody reaction that is probably leading to the rash. There aren't too many studies, but more pretty much the whole scientific community believes that the immune response is associated with the rash. Now, these are some simple findings. Don't have to worry about this. See, what you have to remember is because it can affect all the mucous membranes, including the eye, it can cause conjunctivitis in the eye, respiratory infections, complex spots, I told you. Okay, so this part I didn't tell you. Okay, this is something also I have to explain to you. This is in virology, this is a common term. It's not common in other microbial infection. So what is this term? This term is called pathognomonic. Again, I'll tell you, pathognomonic is a term. What is the meaning of the word pathognomonic? It means highly specific. Now, I'll qualify that a little bit. When a clinician sees complex spots, he can be almost 100% certain that this child has measles. Okay, when a child has maculopapular rash, but does not have complex spots, the clinician may not be able to rule out measles, right? So absence of complex spots does not mean no measles, but the presence of complex spots almost confirmatory for measles. Do you understand what I mean? Or anybody has any questions?
No, such
It's clear. Okay, so it's highly specific but may not be very sensitive. That's the meaning of the word pathognomonic. Okay, there are some simple things that there are 1 or 2 viruses for which this term is important, and measles is one of them, and the rash is maculopapular. Anybody who wants to guess what is leukopenia? The word is self-explanatory. No?
Decrease in number of leukocytes.
That's correct. It's decrease in the number of white blood cells that we have, because measles can also infect white blood cells. Remember, the first receptor that it uses, sorry, this one, the SLAM, affects T cells and B cells, so it can actually cause a decrease in the number of white blood cells. That's important to remember, because I'll tell you why this is important before today's lecture, before the end of today's lecture. In general, most of us, if we get infected with measles, we recover well. Okay, then why are we talking about this virus? Because there's a vaccine, and if we recover well, why are we talking about it? We're talking about this virus because it can lead to a variety of complications. It's not as simple as we think. Although most people recover, there are some unusual complications that are associated with the measles virus. And that's why they even developed a vaccine in the first place. If it just caused something like a mild respiratory illness and coughing spots and a rash, and it resolves, nobody would have had a vaccine for this. Of course, the very fact that there is a vaccine for measles virus suggests that there is more to this than just respiratory illness or a rash. There's much more to it. What are the complications associated with measles virus? This is a straightforward one. You can expect it. It causes pneumonia or inflammation of the air sacs of the lung and make them less functional. So you'll have less oxygen, right? That's pneumonia. Okay, it affects the air sacs, the lower respiratory tract. All of you know the term pneumonia, thanks to COVID again. Now, all of you know the term secondary bacterial infections. Can you tell me where we just looked at it recently, and what is the reason for that?
So in influenza infection due to less mucin in respiratory tract.
And also it just takes away the top layer with the cilia, right? It damages those cells, either the virus itself or the immune response, damages the cilia or the top layer epithelial cells containing the cilia inside your respiratory tract. So the mucociliary clearance is reduced. So you have a higher chance of secondary bacterial infections there. Here you have a higher chance of secondary bacterial infections, primarily because this virus can also affect your T cell and B cell function. We're going to also look at it in a bit. So regardless, actually, it can cause pneumonia. This is viral pneumonia caused by the virus itself, the measles virus, often abbreviated as MV. But it can also cause what we refer to as secondary bacterial infections. Now, I told you it can affect the eye. It can also cause corneal ulcerations in the eye, bacterial infection of the eye. There have been at least a handful of reports that have documented blindness following serious ulcerations after measles, but this is not common. So, see, this first two complications, it infects the lung, causes pneumonia, secondary bacterial infections. It causes ulcers of the eye and secondary bacterial lesions. Now, we are going to go into the more important complications of measles. Now, the first one we are going to look at is something called post-infectious encephalitis. Okay, what is post-infectious encephalitis? As the word suggests, it's after the infection is resolved. It's post-infectious. That's what this word exactly means. After your body is able to clear the virus, you have a disease, you have inflammation of the brain, that's called encephalitis. Now, why does this happen? This is more like an autoimmune response to the myelin basic protein. Now, this myelin basic protein, can anybody tell me what is myelin? What is the function of the myelin sheath in one simple line?
So it provides insulation so that the impulses don't go haywire.
Perfect. Actually, these are the myelin sheath is present around. It just insulates the nerves, all our nerves. And the function of the myelin sheath, like one of you rightly said, is to insulate the nerves in a manner that the nervous impulses that travel through the nerve, they're not lost. It insulates and keeps the signals intact. That is the function of the myelin sheath. If there is no insulation, by the time your brain says to my hand, flex my hand, it will be lost in transit. So this is what prevents the loss of signals when nerve impulses travel. This is very important, the myelin sheath, and it's made up of, this is an important component of the myelin sheath. It's called the myelin basic protein. Now, what does influenza have to do, what does measles have to do with the myelin basic protein? So what happens is when we get infected, we produce antibodies to a variety of epitopes. of the virus, even one protein, I've told you several times, will have many, many epitopes, and our body produces antibodies to all possible epitopes if they are immunogenic, if they can elicit an immune response, our body mounts a strong immune response. So there will be thousands of different types of antibodies that our body produces against measles virus. That's also true for example, if you get infected with influenza virus also, there would be at least several hundreds of antibodies that will be produced by our body against influenza virus, for SARS-CoV-2, everything. Because even though they don't have hundreds of protein, each protein has multiple epitopes. So we have like several hundreds to several thousands of antibodies are produced against any virus infection. Now, some of those antibodies, depending on the genetic makeup and the presence of other cofactors in the individuals, what they can do is they can go and target the myelin basic protein, which is the key component causing insulation. So as you can imagine, this is an antibody induced disease, and it, like fortuitously or accidentally, the antibody that your body produces to protect against the measles virus turns out to be bad, because in some individuals, very small proportion of individuals, it turns out that some of these antibodies can go back and target, produced against measles virus, can go back and target the myelin sheath or the myelin basic protein within the myelin sheath. And then it leads to the deterioration of the sheath. And this happens 1 month after the infection, which means basically in most cases, you will not have any virus. Happens only in about 1 in 1,000. That's a good thing. The bad thing is, if it happens, there's a 1 in 5 chance the person may die. In some individuals, the 4 out of 5 who survive, almost half of them have some neurological complications because there's been some damage done to the myelin sheath. So this process of the body's own immune response to a microbe causing an autoimmune disease is present in many viruses, many bacteria. It's not uncommon. Anybody wants to give me a word as to what this phenomena is generally referred to, or a blanket terminology for this kind of phenomena? Guys? Nobody?
So not sure, but is it sepsis?
No, sepsis is something different. Sepsis is actually,
Autoimmune response.
this is autoimmune response, but there's another terminology.
So cross-reactivity?
Cross-reactivity, true. Okay, so maybe I'll just tell it. So I think many of you may have heard about it. Maybe I didn't ask the question right. So the terminology that's used for such a phenomena, usually when a body's immune response to a microbe ends up damaging your own tissue, that phenomena is often referred to as molecular mimicry. So this is because some protein of measles virus is very similar to a particular epitope of myelin basic protein. That's why this happens. No, that's okay. It's not sepsis, like one of you said. Sepsis, the concept is very different. Sepsis, what happens is some component of a microbe, usually the cell walls of bacteria are the best studied. Even SARS-CoV-2 can cause something like sepsis. Let's keep that aside. But for example, the cell wall of bacteria, when they are present in the blood, or when they spill over into the blood in large quantities, or when you have critical amount of bacterial cell wall in the blood, although you may not have live bacteria in your blood, the cell wall in the blood, large quantities, is enough to trigger a very acute, strong immune response, and which is very bad for the body, and that's called sepsis. You don't necessarily need live bacteria to cause sepsis. If you have enough bacteria, if you have some bacteria and they grow, and bacterial contents spill over into the blood, not necessarily bacteria, especially the cell wall, for example, that can cause sepsis. Anyway, that's besides the point, because some of you brought it up. So this is post-infectious encephalitis. Okay, so all of you got this, PIE, and why it's called post-infectious also, I hope all of you understood that. The second complication that's associated with measles virus is called measles virus inclusion body encephalitis. So it's a long word, but I'll tell you very simply. It's referred to as post-infection, but these hosts are immunocompromised hosts. Typically, it occurs in immunocompromised hosts. They are not able to fully clear the virus, and gradually it starts affecting the central nervous system. It affects a variety of CNS functions, and usually these individuals succumb to it in about 3 years. The last neurological complication, again, this is an important one. Like all of these are important in terms of complications. So you see, it affects the lung, the eyes where it infects, but then the brain has 2 or 3 complications. One is PIE, the second one is this. The third one is called subacute sclerosing panencephalitis. Then before I go on to what this is, I'll tell you when this happens. Let's say I get measles virus infection, and if I'm going to get this, it can happen to me like even 15 years after I get infected with measles virus. That's why it's called subacute. It's very slow. It happens several years, like up to 15 years after getting the infection. Also, this can happen. That's why it's called subacute. It's called sclerosing because there is damage to the brain, and then there is some sort of damaged regeneration that causes scarring. And this is just inflammation. All of you know this. That's the name. What happens here? So here, it's more autoimmune response to this protein, accidental. Here, that the host is not able to clear the virus and the virus persists. That is why this happens. Here, it's a different reason. Here, what happens is this virus in the brain, but what happens is it's a defective virus. Now, what do I mean by defective virus? Measles virus makes a bunch of proteins, but from patients with subacute sclerosing panencephalitis, or SSP as it's called, usually you'll never be able to isolate the virus from the brain. Pretty much whoever has tried has failed to isolate any measles virus from the brain. But most of them found measles virus proteins, especially some proteins that are present, some proteins are never present. The matrix protein was never found. Other viral proteins were found. So which means that the virus is there in the brain, but it's defective. It's not able to make all of its components to assemble and replicate. That's why you're not able to isolate the virus from the brain. However, the proteins that are present, they are able to accumulate and cause damage inside the cells they are present in. And this happens very gradually over a long period of time, and it's very rare, 1 in 10,000 cases. So basically, this is almost always fatal. That's where it ends up. The good part is it occurs in a small fraction of population. If we did not have a vaccine, pretty much all of us will have, the whole world will have measles. It's before influenza, this was the most contagious disease known to mankind, measles virus. After that, we knew influenza is also highly contagious. Now we know SARS-CoV-2. So these are the 3 top ones that we know of in terms of how contagious they are. So if you look at why this is important, is without the vaccine, 1 in 10,000 would be a very large number if you think of the whole population of the world. Okay, so this is just a cartoon of the virus. The last complication that this virus can cause is that it can cause temporary immunosuppression. Does anything that we looked at so far ring a bell in terms of why it could cause immunosuppression?
So the removal of WBCs.
Yes, that's what I told you. Yeah, so I'll perhaps remind you of where we started. Go ahead.
The receptor, sir, SLAP receptor.
This is also very important. Very good, actually. So two of you have brought out 2 very critical points, and basically what we need to understand in terms of the basics of why measles virus can cause immunosuppression. These 2 are the most relevant points. So we'll go on. So one of you said SLAM receptor. So this is a signaling receptor that's important for activation of T cells and B cells. Is present on T cells and B cells. And what measles virus does is there are some proteins of measles virus and it makes these non-functional. So there is less activation of T cells and B cells that suppresses the immune system temporarily till the measles virus is cleared out. It is actually, it's not functional, fully functional. Some people call it, some people have even documented that measles virus leads to amnesia of the immune system when we have the infection and we gradually recover. So this is one of the reasons, because the receptor is present on T and B cells and it inactivates something that is essential for or necessary for activating B and T cells. Measles viruses affect that. That's one reason. The second thing is because it can affect immune cells and it can cause a decrease in their number. So that is another reason that it can cause. So both, and that is what I meant by, I'll explain the leukopenia or decrease in WBCs in a bit. This is what it means. This is a very important complications of measles virus. For about 4 to 5 weeks after you get infected, your immune system works a notch below what it's supposed to work, and it takes about 4 to 5 weeks to recover back to the full strength. Now, the reason is it infects lymphocytes. It can modulate cytokine expression. Cytokines, if I didn't tell you what they are secreted substances. They are usually secreted by immune cells. Mostly they're secreted by immune cells, and this is and they can travel to other parts of the cell, other parts of the body, and activate or suppress the way we mount an immune response. They are immunomodulators. Interferon is also a cytokine that's secreted, right? It goes and binds, tells the other cells that the virus is around, right? So this is also an example of a cytokine. Measles viruses can suppress cytokine expression from T cells and B cells. So it suppresses cell-mediated immunity, which is the arm that is responsible for killing virus-infected cells. It can also affect humoral immunity, because B cell activation is also suppressed to a good extent. So it can affect T cell and B cell immunity. Okay, so that's what I have in this slide. How do we know this? How did we come to know about it? Because scientists and clinicians about 50, 60 years ago, they started noticing that whenever people have measles, at that time measles was so common that almost every day a clinician would be able to see measles case. So what they noticed those days, that if somebody had measles, they have a reactivation of herpes virus, or they have reactivation of tuberculosis. Now I want one of you to try and tell me in one line, based on whatever we've looked at in this course so far, what this could mean.
So because of immunosuppression, the herpes virus and
That's correct. But that is very true. But what do we mean by reactivation? Nobody? So what it means is
latent infection, like they remain in hiding. So when our immune system is suppressed, so they get a chance to
So
multiply.
microbacteria.
Say that again. Whoever spoke last, can you say that again? I heard everything before that. Okay, I don't know who spoke, but if you want to say that again, you can. Please go ahead. No, okay. Okay, whatever. I think you're on the right track. So I don't know if you recall that I said if you got chickenpox infection.
The second infection,
Your voice is breaking a bit. I don't know if it's just for me or if it's for everybody else. Okay. I couldn't hear you very clearly, but doesn't matter. I'm going to recap that for the whole class. I don't know How many of you remember when we started the first lecture? I think it must be in the first lecture, I believe. I told you that viruses can cause acute self-limiting infection, which means you clear it within a few days, no problem. Then they can cause chronic infection, which means you're never able to clear it. That's example is HIV. Then you have infections where there is latent infection. When we say latent, that means the virus is not actively replicating. A good example is chickenpox that many of us have had, or there are other examples also. So a virus called Epstein-Barr virus. Like everybody sitting in this class has been infected by the virus. You may not know it. It causes only some mild fever kind of thing. We all had it before 5-6 years of age. Most of us have been exposed to it. Pretty much the whole world, not just India. Okay. So, and this virus also hides in the body. So these are all mostly herpes viruses, which can hide in different parts of the body. Some of them choose neurons, for example, chickenpox virus. Some others choose some WBCs to be latent. So there are latent viruses. So, for example, the entire human population has, any human being that you take will be harboring at least 2 to 3 herpes viruses would be hiding inside their body. They got infected, they cleared it. The with which can actually which have the virus genome, but it's turned off its replication. It is waiting for the right time to get reactivated. And these viruses which are latent, they have sensors which can sense the extent of immune response. And when they sense there is a temporary immunosuppression, they turn on their replication again and start replicating. So that's called reactivation. Among bacteria, the only one that generally causes latent or chronic infection, the most well-known one and one of the very few among bacteria is tuberculosis. So like 1 in 2 Indians, 1 in 2 of you sitting in this class, we have tuberculosis hiding in our macrophages or somewhere, and we are not diseased. But measles virus infection can allow the tuberculosis to be activated, because again, the Mycobacterium tuberculosis can sense that your immune response is suppressed, and it finds an opportune moment to start replicating itself and propagate itself. So this is called reactivation. Now, we have another 10 to 12 minutes. We'll finish this. So there's something called atypical measles or modified measles. What is this? This is people with partial immunity, especially actually mothers, when the maternal antibody, when a newborn is born, the child just after birth has antibodies from the mother's blood. So this antibody, if this infant gets exposed to measles virus, because it already has antibodies against the measles virus, which it acquired from the maternal blood, this infant will have a modified kind of measles or atypical measles, where it doesn't take 1 week, but it may take 2 weeks or 3 weeks for symptoms to appear, because there is more, the antibody reduces the number of viruses available, increasing the incubation period. Symptoms are milder, no public spots, milder rash, all this. So because there is less virus, there is milder rash. So this is called modified or atypical measles. This may happen because of maternal antibodies circulating in the newborn against measles, specific antibodies against measles. Now, there is only one serotype of the measles virus that is there. Usually, if you get infected, if you had it, usually you'll have lifelong protection. It's very contagious before the rash, because a large amount of shedding happens before the rash. And that time you don't know it, because you might have just a very mild cough or something like that, but you're shedding tons of viruses because it's there in your lungs. So only after you have the rash, most people realize they are infected, but the spreading is done or the damage is done well before that. And another thing is that almost everybody who's infected with measles, they are usually symptomatic. Usually the proportion of asymptomatic infection is almost close to 0. So when did we first develop a vaccine for measles virus? By 1960s, the vaccine was developed and introduced somewhere in the mid 60s. You can see this is all hundreds of thousands of cases. Okay, this is not the number. You have to add 3 zeros to it. So you can see that after the vaccine was introduced, the cases almost dropped to a flat line in the United States. By 2000, America declared they had eradicated measles. Okay, that's a good thing. But in 2015, 15 years after they said they've eradicated measles, suddenly over 100 cases appeared in a Disneyland outbreak. Now, anybody wants to guess how this could have happened? Or why this could have happened? Guys, nobody wants to guess?
Some sort of mutation.
There's a mutation.
Okay, if I tell you there is no mutation, anybody wants to guess? You want to focus on this, you'll guess it.
For international visitors.
That is correct, right? It's eradicated from America, it's not eradicated from the whole world. But then if all Americans are vaccinated, this should not have happened to this extent. So I'm going to talk about, in the next 5 to 10 minutes, I'll talk about why this happened and some other interesting facts about vaccination. Okay, so this happened in 2015, more or less recently, 5-6 years ago, a bunch of cases. So I want you to look at this cartoon. This is 1998. This is the coverage of vaccination in different parts. So you can see that gradually it's dropping. It went to very low. Gradually it's dropping, it went very low, and then it came back up a little bit slowly. So what is the reason for gradual drop in the number of, this data is from America only because we don't have very good data on this, and only like a handful of countries in the world maintain very good record like this. So anybody wants to guess why this dropped so much from 1998 onwards? Any guesses? Anybody has heard of this group called no-vaxxers? Yes? No?
Yes, sir.
Yes, sir.
Yes, sir.
Okay, so this group is it's a large population in developed countries, especially America, who call themselves no-vaxxers. And this is the mother of all those people. This paper in the Lancet in 1998. So what did this paper say? This paper studied only 12 children. I don't know if you can see the font size. They studied only 12 children totally. And they conclude that some of it, I don't know if you can read this, but in the slides, you'll be able to read it. They conclude that vaccination may be associated with developmental regression, or basically they associated with autism spectrum disorders, where children lack development in one or more or all aspects. So that is what they did, but just by studying 12 children, they conclude this. And this paper had over 2,000 citations. That means it's cited by so many papers, and it created a flutter in the whole community, not just scientific community, but also laymen. They started thinking
I
that
guess it was a British physician, right? Andrew Wakefield or someone.
yeah, that's right. This is the Wakefield study. You're right. This is the Wakefield study, and it is British. You're right. And this actually changed the way we looked. Like everybody looked at vaccination, especially common man. So this paper was essentially retracted after a few years, but it has done the damage, and this was the primary cause for this group called no-vaxxers. Now, this is just, you don't have to read any of this. This is another top journal like the Lancet. You can see this is 4 years after that, 2002. And this study concludes that there is no association between vaccination and autistic disorders. So this study was published in the New England Journal of Medicine, a top journal again, but if you have two studies like this, usually people would try to go by whatever is the safest one for them. Not vaccinating may be better, even I personally know some non-vaxxers actually in the US. So it's not uncommon. It's a very common thing in the US. Now, what is the problem with this? The problem is that the 125 cases that occurred in the Disneyland outbreak, if most of the US population is vaccinated, that wouldn't have happened. So although vaccine coverage is slowly improved, it slowly improved after that. It's not 100%. The no-vaxxers actually are the problem. So they still remain susceptible to infection because they are not vaccinated. If somebody from foreign, overseas country where the coverage is poor brings in measles, all the no-vaxxers in US can get it, although they have very good coverage and they don't usually have any cases that originate there. It can come from abroad. It can also, no-vaxxers from America who travel outside and come back can bring it back to the US. So it's important to vaccinate the kids, and it's very clear to the scientific community that at least these vaccines don't have anything to do with autism. That part is now very clear, but the damage is done that some people have it strongly ingrained that they cannot change their opinion. Now, how do you detect measles virus in the laboratory? Very simple stuff. Respiratory virus, respiratory samples, you can use conjunctival samples. It's shed in large quantities in urine also because it affects all your epithelial cells or almost all the moist parts of your body. Immunofluorescence was the mainstay. Now we're slowly moving to RT-PCR. You can also grow measles virus in cell lines and then confirm it by immunofluorescence. All of you know what immunofluorescence is, using a fluorescent tagged antibody to identify antigens that are present in the measles virus. For measles virus, actually, serology is also a good diagnostic tool. You can look at IgM class antibodies against the measles virus that indicates you're having an acute infection. Now, so far we've looked at polioviruses, enteroviruses, Coxsackie echoviruses. We've also looked at influenza viruses, but I generally told you serology is not good, but here I say serology is good. Why is that? Guys?
There's only one serotype.
Perfect answer. Actually, there's only one serotype, so that is a very good thing. And we get vaccinated for measles only in childhood. So usually most of the population will not have a vaccine-induced antibody or anything, which will be IgG class only. See, I don't know how many of you know this. Whenever we get a vaccine, I'll just put a vaccine or an infection, a natural infection. The first antibody that's produced in the body that in the first 10 days is IgM class antibodies against the virus. Within about a month, after 1 month, there is something called a class switch, and the same antibodies would be produced from IgG class, and this IgG class antibodies would be present in our blood for the rest of our lives for very long periods of time. So this is what we find due to vaccine. When I got vaccinated at 5 years, or you got vaccinated at 5 years, you still have IgG antibodies against measles virus, but you will never have IgM antibodies because they are short-lived and they are present only during acute infection. They will be produced. When you get exposed to measles virus again, or when somebody gets exposed to measles virus for the first time, that is typically when they will produce IgM class antibodies. After that, there is something called a class switch. It will become IgG. So if you have IgM antibodies that's not induced by vaccine, and there's only one serotype, you can detect just by taking blood and looking for IgM class antibodies against measles virus antigens. You can also say that you're having an acute ongoing infection with measles virus. So the vaccine is a very good vaccine. It's used worldwide. It's part of universal vaccination program, and it's free for pretty much in most countries. There's only one serotype which helps the vaccination. It's combined with, it's called MMR because it's combined with measles, mumps and rubella. So measles, mumps, and rubella makes it MMR vaccine. It's a live attenuated vaccine. This is the measles coverage. America, in spite of the Novaxers, has over 90% coverage. India, although everybody is willing, just because we have to grow up in terms of literacy and other things, we still have only about 60-70% coverage. It's improving. I think we'll get there soon. The vaccine is very good because it's a live attenuated vaccine. It's not given to certain groups. Pregnant women because it can infect the newborns, immunocompromised hosts because this is a live attenuated. You don't know what will happen if you're already immunocompromised. It's not given to them. Usually for normal people, for most of us in this class, we will not need anything. We will just recover. Body will fight it out. There are no antivirals for measles. I think that's all I have in the last minute or two. I just want to say that these measles virus outbreaks, they keep on happening in different parts of the world, and they cost a lot more than we think, because it also has public health implications in schools and other places, especially if your vaccination coverage is not good. This is again just to show that it happens in multiple countries. See, there are many countries. Anytime you Google it, in the last 4 weeks, you'll find an outbreak. So that is all I have, I think. Yeah, that's all I have. Any questions on today's class? Guys, I'm finding you guys are becoming less and less interactive. I don't know if it's
So no
because,
question.
yeah.
So just one doubt here. So
Yeah,
in the case of SSPE, we observed that like 15 years after the infection, the traits start showing. And you explained that the matrix protein is absent in these defective viruses. So, sir, the body cannot detect them and make sure that they do not exist?
Yeah, so it appears like the virus is present only in some cells, and it's usually in the brain. They may not have access to a large number of antigen presenting cells and other things, but these are going to be brain cells, other kind of neuronal cells, not necessarily antigen presenting cells. The second thing is, if all the components are not made, we may not be able to mount a good immune response. See, so even in the like the latent viruses, their genome is usually intact, but they just reprogram their genome not to make proteins, or not to copy their genome until they find an opportune moment, like the immunosuppressed moment. So they can be inside the cell, inside our cells, and remain undetected based on one or two things. Actually, one, if the antigen is not presented, that becomes dangerous for the body and good for the pathogen because it goes undetected. If antigen presenting cells don't have access to it, that is one issue. The second issue is, these viruses are able to modulate the amount of protein production also. It's not intentional. They don't want to cause disease to us or anything. It just so happens that whatever is defective makes the extent of protein production much lower, which allows the persistence. So if it produced, for example, tons of other proteins, what you say would be perfectly true. The immune system would kick in and figure it out. But they produce only small quantities of protein, which are just barely detectable using sensitive methods. So that is not critical, that doesn't cross the critical threshold for eliciting a strong immune response. So that is the reason why they're not able to do it. And nobody is able to isolate a virus. They found virus proteins. And the theory is that it's just aggregation of small quantities of virus proteins that leads to cellular damage in the brain. Does that answer your question?
Yes, sir.
Okay, guys, any other questions you have?
So I had one question. So how do these split vaccines work, like vaccines which vaccinate for more than one kind of disease?
They work very well. There are many vaccines now. MMR is one example. Is that your question? Multivalent vaccines for multiple viruses together. Is that your question?
Yes, sir. So like how are they designed basically? That is what.
Okay, so see why it was designed like this. All three viruses cause fever with rash, and they were all a big problem in around the same times. It started spreading throughout the world, and they were designed together. Now, why are they designed together? Can you add something to it? I do. Basically, even if you add, for example, SARS-CoV-2 virus to that and make a vaccine which is 4 viruses or against 4 viruses, that'll absolutely work. This is one example. The only thing I don't know is how they are combined. That's by trial and error, I believe. And these 3 were combined for a particular reason. They all cause fever with rash. To answer your question about how they work, usually there are enough studies that show that if you have multiple vaccines together, if you give it to an individual, there is no decrease in immune response to any one individual component. It's just as good as getting them separately. So because of that, multivalent vaccines exist. This was the first one. Now we have actually, I think all of you also have had DPT, diphtheria, pertussis, and tetanus.
Yes, sir.
Right? And now that's coming as, I think one more thing is added to that. Now that's also a quadrivalent vaccine. So this is not uncommon to have multiple pathogens combined and have a one vaccine against multiple pathogens. So usually there is some similarity. Here the similarity is the disease. In DPT, mostly the similarity is all of them are related to toxin-mediated diseases rather than the bacteria itself. They are all related to toxins. They are more like they serve as antitoxin vaccines. So something leads them to be combined. But basically the short answer is you can combine other things also. It will work just as well. We don't do it just because we don't want to experiment like that on human beings. But most likely that will also work.
Fine, sir.
Okay. Okay, guys. Thank you. I think we overshot by a minute. Okay. Bye-bye. Thank you.
Thank you, sir.
Thank you, sir.
Thank you, sir.
Guys. Thank you, guys.